We talked about all the drugs that target viral entry all the way to the viral exit. Okay, so let's talk about antibiotic resistance. Okay, so when we talk about resistance, we're talking about the lack of effect of an antimicrobial agent, a previously sensitive microbe. Okay, so we do have spontaneous mutations. All right, spontaneous mutations happen from anywhere from 10 to the 6th to 10 to the 9th base pairs. Okay, but we've got 3 other ways. What are the 3 ways that we move genetic information in bacteria? This is from
transformation, transduction.
Conjugation, transduction, transformation, right. Okay, and then we've got spontaneous mutation. So this is how we get antibiotic resistance. So when you see that R at the top, that's for resistance, okay? All right, so let's talk about what are some of the specifics of development, okay? So altered enzymes. The drug target is no longer affected. So what happens? The bacteria realize, I mean, this is not a conscious thing, I'm not sure if they're conscious, but they have a target that's being used by an antimicrobial, change the target, mutate, so you use a different enzyme or you have a different protein. Okay, membrane permeability. All right, these bacteria, they have pumps. So as the antimicrobial comes in, they actually synthesize pumps and put it on their cell membrane and start pumping the drug out. Okay, yeah, they're pumping it out, and so this is the primary mechanism of tetracycline resistance. Okay, isn't that clever? I mean, they're so clever. What doesn't kill you mutates and tries again, right? Okay, so this is the antibiotic degrading enzymes that inactivate the drug. Penicillin does that. We saw that with beta-lactamase, right? Okay, we have an alternate synthetic pathway or new enzymes that come up. Okay, so how does this all happen? It's mutations or exposure to these resistance genes. When you see resistance genes being transferred from bacteria to bacteria and bacteria to different bacteria species, anything can happen. Anything can happen. Okay, so we talked about spontaneous mutation rate. It's tiny, but if you divide every 20 minutes and do binary fission every 20 minutes, 10 to the 9th becomes significant. So you can have new variants come up pretty quickly. Okay, how does this this is a map of who shares genes with who. Okay, so E. coli can go back and forth between Salmonella. Neisseria can only go to E. coli. E. coli can't go back. So pay attention to which way the arrows go. Okay, Pseudomonas can go out to other Pseudomonads and Proteus, but Proteus can't go back to Pseudomonas. Okay, so there's a lot of genetic exchange. So you could get genetic resistance genes from Haemophilus that ends up in the Pseudomonads. Okay, yeah, so there's a lot of gene exchange. Okay, and that's not even so right now your microbiota is exchanging information. Anytime you take antibiotics, those that are resistant that stick around will exchange information from your microbiota. There was a study that was done at Emory that showed that infants that are 6 months old already have drug-resistant E. coli in their microbiome. 6 months. Okay, you think about a 6-month-old, they're usually home, they're not going out, they're not clubbing, they're not going to school, right? They might be going to daycare, but you know, that's awfully young to be having drug-resistant E. coli already in your gut that is detected. And that's data from about 7 years ago. Do you think the COVID pandemic increased or decreased microbial resistance? Increased. Why? So many drugs. So many drugs just given. And, you know, when you look at the strains of COVID that are most resistant, who are they in? Not the healthy people that recover quickly. It's in the old that survive, the very young, and the immunocompromised. Some of these really aggressive strains of COVID were thought to come from immune-compromised patients that were in care facilities for months. Well, that gave that virus time to mutate, right, within itself. And so, yeah, so kind of same thing here, all right? Okay. Oh, so I got a great email questions last week, and I thought, ooh, I wanted to think about it. I've asked a few folks about what their thoughts are, and now I'm going to ask you. All right? If antibiotic resistance is such a challenge and such a threat, why do physicians prescribe so many antibiotics? Money. Okay, explain that one to me.
Well, they're getting paid.
Actually, they don't get paid. No, they don't get paid. There's only one specialty that does that, and that's oncology. So, yeah, so your primary care physician doesn't matter, right? Doesn't matter, they're not getting paid. So that's not probably the best reason. Okay, so but why would the physicians prescribe them? If they know it's such a threat, why do they prescribe them so frequently?
Because it's extremely effective. Like at getting it out.
And what?
I like getting it out.
Getting what out?
Getting the microbe or getting the.
Yeah, but why? I mean, you see a lot of. Why are they overprescribed? If you know your patient can survive and they just have to fight it, why would you prescribe it, or why would you prescribe it for a virus? Yeah, Raphael.
Oh, it's a misconception from the community. So the community has this idea that most antibiotics is working, so doctors would assume that their patients would think that they're lacking care by not prescribing antibiotics.
Okay. Okay. Yeah.
Could it be that they just don't want patients, they want to get patients out of the hospital? Like, because if somebody was like, ended up.
Or out of their office, right?
Yeah.
Oh man, not that patient. Okay, yeah, here you go. Could be, yeah.
Maybe to prevent the spread, like if maybe that person is gonna have side effects, but the spread is gonna end right there.
Okay. Yeah, no.
Well, maybe just to ease the patient's symptoms and just give them something.
Give them something. So let's say you have a patient that comes in and is pretty viral. They still give antibiotics. Yeah.
I had an experience with my daughter. She had a little bump here, and they put her through 3 rounds of antibiotics before testing or doing a biopsy. So I gave her 2. The 3rd one I didn't, and she was referred to a dermatologist. And they finally biopsied it, and it was not an infection. It was nothing. It was benign, but they put her through 3.
They were gonna put her through 3 rounds.
Yeah.
Yikes. Yeah.
Is it like a placebo effect thing? Where it's like the patient's like, oh, this is gonna help me feel better, and then it's like doesn't really actually do it, but they trick themselves.
Yeah, but I'd give out Pez, you know, rather than that. Yeah.
It's because the antibiotics are like a wide spectrum. Cover a whole lot of
Yeah, but if you aren't sure you've got something that can be treated by antibiotics, why do they give them out? Yeah.
I don't know if it is just logical as heck, but help the immune system such like how the microbes can also pick up genes and such. Could the immune system also do that?
No. Good idea though. That's very creative. I like that. Yeah. Yeah.
I genuinely think that it is like just to get you out of your office because like my son was sick for over a month and I took him to the doctor so like 5 times and he wasn't getting better, and then the last day, so finally his doctor to the doctor again like 2 times after that, and to see like to get a second opinion, and then finally I made them like do tests on him to see if he had a virus, because I wasn't gonna give him antibiotics, it was just a virus. And then they did the test, and he didn't come out positive for that or anything. And then I gave him antibiotics, and he did get better with that.
Oh, so you had some kind of infection.
Yeah, but I didn't give it to him until like.
You'd exhausted everything
Yeah,
else.
so like his doctor was like sick of me, and she was like, "Here, like just give it to him." She was like, "I'm not gonna give him antibiotics if he doesn't need them." And then she finally did all the testing, and then I decided to give it to him.
Okay, that convinced you.
Yeah.
Okay. That's a great question. This is a great question. Yeah. Yeah, and some folks in the population, you want to do that. You know that, okay, you got a patient that's got, somebody that's got CF at home, cystic fibrosis at home. I would treat that patient with antibiotics because I don't want him passing anything to someone who was home there. Or if let's say that they don't, they're immune suppressed for some reason. Yeah, I can see doing that. But generally, I think you hit it on the head. They want you done, right? They get, how many minutes now? 6 minutes per patient? Yeah. And look how long it was before they even cultured or even biopsied.
5 months.
5 months, yeah.
So why don't they biopsy first? Because wouldn't it be less expensive to biopsy than to give so many different rounds of antibiotics first?
Well, the biopsy, the medical group has to pay for. You got to get a dermatologist, you got to do path, you got to do that.
Or like even cultures.
Yeah, that's still. But if you give the patient antibiotics, that they don't have to worry about that. Their insurance isn't covering that. So.
I think also it has to do a lot with where we are. Like, I'm Salvadorian, so first, the third world countries, like, I feel like it's cheaper to give them medicine, even if they're strong, than doing all the labs. They don't even have the resources sometimes.
Yeah, exactly. And getting into a dermatologist to do that takes months anyway, right? Wow. Yeah. What do you think? This is crazy. Yeah, I had the same conversation with my docs this weekend about why do you do that? And I have one that's like, I don't do it. If it's not cultured, if it's not that, I don't do it. But he also works for the VA. A little different. Okay? But those that are in practice and not in the VA, they tend that they said if we're 80% sure that this could be bacterial, we will give it. And they'll just give some broad spectrum. And that's why this is going to get worse. So, isn't that crazy? Yeah. Ooh. So somebody in here needs to go into research so we can get some better antibiotics. Because we're out. Yeah. Good times. Okay. Which brings us to this slide. I think this slide is fascinating. Okay? Antibiotics that don't work on colds. Like unnecessary antibiotic prescriptions. So, if they're unnecessary, this means most likely this is some kind of virus. Look at that, 30% of ear infections. You know most 30% at least are viral. They'll go away. Now, if you look in the ear canal and you see all this pus coming out and little elves and stuff like that, antibiotics might be a good idea. Okay? Common cold. All right? Don't give antibiotics for the common cold. By the way, wow. There are colds going around. 2 families of colds going around right now. And it's estimated to peak by the 3rd week in May. So if you got a cold, you got any, I don't know if you've noticed it, a lot of colds, okay, yeah, they're out. This is their normal cycle. They tend to like springtime. I don't know what this weather is. All right, bronchitis. Here's a good one, 80%. Why? Most of bronchitis is what? Viral. Yeah. Sore throats, 50-50, sinusitis, 50-50. Okay, so here's a question for you. Sinusitis, all right? You got a cold, you got bad sinuses, it's been 3 or 4 weeks. Do you get antibiotics then usually? Yeah, because why? The virus is long gone. what has happened is a virus destroyed your tissues in here, the mucous membranes, and some bacteria have gotten in and you're not feeling better. It's very unusual to have a sinus infection for like 4 weeks, okay? That's 100% viral. Usually don't see that unless what? Old, young, immunosuppressed, right? Okay, cool. All right. Okay, so got things here to fill in. Inappropriate usage, exposure, suboptimal exposure, suboptimal levels of the drug, exposure to microbes carrying the resistance genes. And how do we notate the resistance genes? With that capital R. Yeah. Okay. I like this. Okay. What kind of curve is this up here? It's a bell curve, right? We see bell curves a lot. I like this because it kind of shows the progression once you give antibiotics. So here we go. Here it is. We've got all of these 3 organisms in you right now. Okay. By day 3, what do you notice about the curve? It's shifted to the right. So the sensitive ones are disappearing and the intermediate ones are disappearing. These have not been touched yet. By day 6, we're pretty good. Still haven't gotten that. But by day 10, we've gotten the numbers low. Now, why didn't they show them completely removed by day 10? Because usually they aren't. But here's what happens. Once you give antibiotics here, okay, your immune system has a pretty good shot of doing it. First of all, it's day 10. And as we'll talk about when we talk about the immune system coming up, it takes about 21 days to get a really good immune response going, okay, for something that you've never seen before, okay? So at least you've got low levels. That's okay, because at least we've given the immune system a chance. Does that make sense? Now, what happens if you stop at day 6 because you're feeling good? Who's left behind? Boy, those resistant ones, okay? And they're going to spread to other hosts. They're going to spread all over to all your friends and family, okay? It's causing drug-resistant infections. Yeah, good times. Okay. Any questions about that one? Okay. So, what are the consequences that we see? Multi-drug resistant TB. Okay. Infections that are resistant to any antibiotics that we have. Okay, this figure is about almost 10 years old, not quite. Do we have multi-drug resistant TB in LA County? In Riverside County too. Okay, so anytime you've got a major metropolis, you can expect drug resistant TB. Why? Because people travel. And what population is notorious for carrying TB? A lot of unhoused people, and a lot of unhoused people travel. Okay, it gets hotter than heck here, they're going to go somewhere else to another city. And how do they get there? Yeah, they bus, usually the buses. Yeah. So, yeah. So this is going to increase the cost of treatment, no doubt. Okay. Now, we've got a number of pathogens like MRSA and vancomycin-resistant E. coli, VRE, okay? And you can see the abscess that you get with MRSA infections, okay? That's a little abscess that comes. I've seen them much larger than that. I'm sure you have too, okay? And once you're colonized with MRSA, does it ever go away? No, okay? I'll give you an example. My dad, before he passed, he had a terrible kidney stone. Terrible kidney stone. So they said, okay, we're gonna go in and sonicate him and bring him back down in the cage and you'll be fine. And they screwed up on somewhere and he bled out. Okay, so he's in an ICU. He's getting packed cells. They're getting ready to intubate, all this stuff. He lived through this, by the way. I mean, he's a tough 82nd Airborne. He was pretty tough. So anyway, he lived through all of this, but guess what he picked up between the ICU and the care facility? He picked up MRSA, right? He picked up MRSA. So once he was colonized by MRSA, there were a lot of things going on. You had to let your dentist know that you're colonized by MRSA. Had to let every physician know. If anything showed up on his skin, suddenly, what did we do? It was a trip to the urgent care or to the doctor if we could, to make sure that it didn't come back, because he was older. I mean, at this point he was what, 82 or something? So yeah, once you're colonized, you're colonized. And you might not have it for years to come, but you get run down enough, what happens? Comes right back. Okay. So, yeah. All right, we're gonna stick to no cartoons today. Today is just like, bleh. All right, what can be done? We could develop new antimicrobials. We can give alternate therapy like viruses. Okay, I'm gonna show you a really cool book. Okay. This is a book. You wanna read something kind of cool? This is a book called The Perfect Predator. I just started it. I had to put it down or else I never would have slept. This is a scientist who did public health. And her and her husband, who was also a biologist, he's a research biologist, went to Egypt. While on their trip to Egypt, he picked up an infection. And it was MRSA, okay? And he went septic, and now he's on ECMO. Okay, so he's really having a hard time with it. He was not improving, of course, it's MRSA. So what did she do? She harvested these viruses that attack methicillin-resistant Staph aureus. And what kind of little critters are these? This is the T2, T4 phages that infect bacteria. He's alive and well now. So this whole book goes through the science of what happened, and then why this was the last resort to save his life. There he is. Wait, let's go back. Yep. So, wow. I would put a picture of this phage in my house if it saved my life too. Oh, I know what it was. It wasn't MRSA. It was a Baumannii infection. It was Acinetobacter baumannii. We talked about that one, right? That's in the soil. Yeah, because they were crawling through caves. So yeah, that's Baumannii right there. Yeah, you bet I would have pictures. I won't put a picture of this, but I definitely put a picture of this. Okay? So if I know you don't have time to read anything or that, but this is really fascinating and well done. Actually, it does. Pretty well done. Okay. All right. What other things can we do? Old drugs. Using old drugs. Just because a drug is old doesn't mean it's not as effective. We're finding, did you know that up to 70% of the drugs on the market right now are used for off-label purposes? 70%. Okay? We're seeing that with GLP inhibitors, right, or GLP-1 drugs, right? We're seeing those. It was used for what? GLP-1 drugs, what are they for? Diabetes. But now, what is their off-label use? Obesity, right? Okay? When you take a Z-Pak, azithromycin. Azithromycin has a little bit of antiviral property. And that's why it makes it good if you're thinking this is a viral plus a bacterial infection. Okay? What was that found? It's an old drug, but they found it actually works pretty well if you get the infection early enough. Okay? So yeah, tweak and improve. And then we have combination therapy. Okay? We've talked about the difference between being additive and being synergistic. Okay, do we need to go over that again? Do you remember that? Okay. No, we don't need to go over it, and no, you don't remember it. Okay. Let's say you give drug X, you get this response. You give drug Y, you get this response. An additive one would put that, there we go. It would be this plus this. This is additive. Okay? What would be synergistic? When you give X plus Y and you get a response like that. That's synergistic. Synergistic means that they provide a response that is much more effective together than would be predicted by an additive response. Okay? Does that make sense? Yeah? Okay, cool. All right, so appropriate use. That's really what we're after, is appropriate use. Okay? Okay, the next two slides are antibiotic reviews. Okay, for you. So give me a second. And we'll transition to this. Yep. Okay. We're going to start the innate immune system. Okay. Everybody's got this calendar down? Okay. I'm going to take a picture of it real quick because I don't have time to write it down. There we go. All right. So I'm going to use this space here. All right. I want to give you kind of some perspective for the immune system, because if you remember when we were doing translation, we talked about how energetically expensive translation is, and what does translation do? It makes proteins, right? It makes the proteins. Okay, so making proteins is the most energetically, thing you can do. Here's the thing. What are antibodies made of? Proteins. Yeah. So do you want, every time you get an infection, do you want to go ahead and make lots of antibodies to it right away? No, you exhaust the host. Okay? You exhaust the host. If you don't have to make something, it's like the lac operon in bacteria. If you don't have to make something, don't. Okay? But if you do have to make it good. Okay? So, all right. So I'm going to give you a little real life scenario. This happened a couple years ago. So every time this year, I have to go to annual training. It's called AT. We don't like it. It's out in the desert. It's hot. Okay? It's dusty. You get sand and dust everywhere you even care to mention. And there's no relief from the heat. When we had this heat wave, I just am like, oh, no. This can't be happening. All right? But it is. Okay. Hopefully it'll cool down. So one of the things that we do to train for emergency responses is we go through simulations, okay? And the simulations are usually in a room about this big. And in the center here, we have kind of a big round table that goes all the way here. You have phones, you have stations, you have things hanging from the ceiling so you know which is which one is logistics, which one is financial, where's medical, blah, blah, blah. Okay, you got TVs all the way around, okay, the room that carry not only feed from the US, but feed from everywhere else. I'm telling you, it looks like a war room in there, okay? And you walk in there and you're given a big envelope and you don't know what the emergency is gonna be, but you gotta take that envelope and you've got to respond and you run the thing. In order to certify, you gotta be able to run the whole thing. So that means you got to keep track of logistics. You got to keep track of things like, fire responses. You need to know everything that you have at your disposal to use. You need to know what your personnel are like, what your air power is like. You need to know what the temperature is like. Is there civil unrest going on at the same time? I mean, talk about Advil. This is one of those things where you need Advil. So I was going in and I had a new lieutenant coming in, okay? And he's handed this packet. And did you guys go to Coachella? Anybody go out to Coachella? Have you been out there? It's hot. It's big polo club, right? Okay, so he goes out, he gets the, he's supposed to be running logistics, okay? And I didn't know this guy. I wasn't sure of his skills, but I'm pretty sure of his skills now. So there's kind of a first response when you have an emergency. So in this one, an earthquake happened and it knocked out the 10 freeway. Okay, and we had massive injuries, we had all these things, but you knock out the freeway, we got a problem, right? Because you can't transport. The only thing left is train and air, okay? So, we're talking about what is the first line that we do, and we sent out a major triage center at where they hold Coachella, because it's a great place and we can land helicopters there, right? Okay, so then there's the next line, okay? When we talk about, okay, now that we've taken care of the most critical, it's hot out here, how do we get food and water in and out, and what resources do we have, and what mutual aid do we have coming in from Arizona and Nevada to help us, and all that stuff going on, okay? So now we get a little more into the response. And then we go to the third level, which is how do you manage this for long term? Just so you know, I still have 2 soldiers out from the January fires. Okay, why? Because they're trying to pay people that went out there, right? So they're the computer operators, right, that are out there trying to pay people. Okay, most people have forgotten about the fires unless your family was impacted, right? When were they? Second week in January, okay? And I still have people deployed for that. Okay, COVID, we finally took the last people out, and that was a mortuary team in 2023. Okay, I mean, we were gone that long. Okay, that's like this, very long, very specific, very targeted. Okay, so let me give you the scenario that this lieutenant had. Okay, all right. So this was out in Indio somewhere so here it is. And you know what? While you're going through all this, things are flashing up on the screens, which are new information that are coming in. And you've got to adjust your plan. Okay? So I'm going to draw some things here. Don't laugh. Okay? So, yeah, I hear it now. Okay? This is a truck. Okay? All right, this is a tractor trailer, and it's carrying hay. I got to put some wheels on here. Okay. All right. So the truck is out here. It's not on the 10. The 10 is down. It's somewhere south of Indio, and it catches on fire because it's hot. What would you do? What?
There goes the truck.
Exactly. Okay, I'm thinking, oh, I know exactly what I'm going to do here. Not this new lieutenant. What did he do? We need some helicopters. This is my helicopter. we get a lawn dart here. Black Hawk. Okay, so here comes a Black Hawk. We always have these little wheels here. Okay, and he ordered 2. Okay, I can't draw the other one. +1. Okay. All right, so he ordered 2 helicopters. All right, where did these have to come in from? They came in from Fort Irwin. And I'm like, Lieutenant, what's your priority in an emergency? Life, property. Okay? Life and property. That's really what your thing is. Okay? We just had the 10 freeway collapse. We got people that are really, really sick that are being brought by private car to the Polo place in the heat trying to get medical treatment. We're trying to get medevacs in. We're trying to get medevacs out. This truck catches on fire. He sends 2 of our resources there. What do you think? Yeah, it's a waste of resources. Yeah, so I'm like, how long do you want to be a lieutenant? You know, no, I'm just kidding. It wasn't quite that bad. I heard it though. You know, like, what are you doing there? Okay, what's the thing to do if you're trying to conserve resources? Because when there's an emergency, and you're going to see this in the clinics that you're at, when there's an emergency, okay, it's not the first 12 hours are awful. But it's a marathon. It's a fast marathon, okay? You can't spend everything here. So, what's the prudent thing to have happen? Let it burn, okay? That driver's safe. I'm sure he got out of the truck. In fact, let's fix it so he does. Okay. Okay, give him a little hat. Okay, yeah, so he got out of that truck. That's great. So the hay burns. You gonna send your fire crews out there? No, they've got people stuck in SNFs that can't move, that need to be moved out and evacuated somewhere else. Okay? So I like this because it's kind of like your immune system. Don't be sending in the National Guard to, to save the hay during an emergency. And when you have an infection, it's an emergency for your body, right? You've got scouts all the time. I mean, when I think of the immune system, I just think of the army, and I'm just so thankful. Because it is. It's like this army of cells that you have that are keeping you well right now. And we know it's working. How do we know this? You're not sick. You're not sick with a fungal infection. You might be sick temporarily with something, but you're a well person. You can do what you want to do. Does that make sense? Okay. I've really gotten addicted to that Reddit on microbiology. Oh my gosh. It's so fun. But there was a guy, a new microbiology student that posted that took the petri dishes and cultured things around his house and then said, uh-oh, am I cooked? So he had this thing, this little petri dish that he'd put out in the air, and it grew 3 fungi and 2 bacteria. And of course, all of us, I didn't say anything, but I'm watching them. The expert micro people are like, that's happening all the time. You've got funguses and bacteria in the air constantly. You're still alive, you're fine. He cultured his phone, he cultured his balls. Okay? Yeah, BALZ. I think I know what those are. Anyway, he cultured those. Some interesting stuff came up on there. Enough said. I would have never published that. Of course, I don't have balls. So anyway, yeah. So I mean, he's like, oh my gosh. And we think back to when we first started micro. What's the only way that you're gonna get sterile skin? Burn it. We don't do that. No, we use all sorts of antiseptic techniques. Okay, but it's really interesting. I mean, you're healthy. You're totally, you're healthy. You can go and do things. When you see somebody that's got a chronic fungal infection, they are not well. Has anybody ever seen that? You will see that, okay? They are not well. They are not well. They got to worry about O2 sats. Oh my gosh. So, but your immune system, is conserved in the sense that you're not going to send out your helicopters for a truck fire, okay, when you don't need to. Why? Because that costs your host. It costs your host ATP, okay? It costs your host a lot. You know how tired you are when you have an infection or when you have really bad allergies? Why? Because your immune system is all ramped up. Of course you don't feel good. And you're trying to repair stuff that got damaged. Does that make sense? Okay. All right. So I want you to think of the immune system responding in 3 kind of phases. Okay? 1, 2, and 3. And that's what we're going to take a look at today, is the innate immune system. Okay? And we'll see what that is in just a moment here. Okay? This is all we do. This is non-specific. Everything that is in this chapter 14 is non-specific. What does it mean? It means that it doesn't matter if you've gotten a stick put in your eye or you've gotten an attack of fungi, the response will be the same. That's what non-specific is. It's not particular to responding to anything. Okay? So, everything in this chapter is non-specific. Okay? So we have all sorts of parts of the non-specific host defense. Let's get right into it. You notice how we talked about the levels of response here? Do you see the same thing? The first line of defense, the second line of defense. These two, these are, oh, I can't even see that. That's okay. Non-specific. Okay? And then this one is very specific. This is where we get antibodies. Okay? What else do you hear of this? This is also called innate. Okay? This one is adaptive, because it's able to change. All right? Now, the key thing here is to understand what each of these first, second, and third line of defense is, and that's what we're going to start with today. Okay, so here's the first line of defense. What's your number one first line of defense? Skin. Skin, skin, skin is your first line of defense. Okay? But I want you to take a look at your hands. Do you have cuts on your hands? Yeah, okay, you do. So do you ever have a complete intact epithelium? No, okay? That's important, because you think about infections like osteomyelitis. Okay? Osteomyelitis, what kind of infection is that? Bone. Okay? Is it possible to get osteomyelitis from a cut on your hand? Absolutely. Absolutely. The bacteria enters from somewhere on your skin, it finds some place on your bone and starts to set up house, right? Okay, so here we go. First line of defense. And then you look at these, the second line of defense. We've got all these phagocytes. What do phagocytes do? Eat. Yes, they eat. They get rid of things. They remove debris. They remove pathogens. Okay? There's extracellular killing. Okay? Right now, you've got circulating a bunch of natural killer cells. And what they do is they identify the abnormal cell and kill it. Okay? They're on patrol right now. Inflammatory response. This is part of the second line of defense. Okay? And we're going to go through the steps of what is needed for the second line. Okay? Fever. Fever is able to what? Increase circulation. Okay? It accelerates movement of cells to areas. It accelerates repair. And also, a lot of microbes don't like the increased temperature. So that's kind of cool how it's got that. Okay? Interferons. Okay? Interferons, they're released by lymphocytes. You've heard of interferon. What does interferon A treat? No, not HIV. But it's one of the viruses. Yeah, interferons are great against it. That was a easy question, right? And then we have the complement system. This is not, oh, I like your hair, I like your nails. No, this is not the complement system. What is the whole point of the complement system? These are proteins that assemble on the surface of the bacteria and punch a hole in it and lyse the bacteria. Okay, that's the whole purpose of the complement system. Okay, these little tiny proteins, there's about 15 in the cascade, they assemble onto the surface of the bacteria and then punch a hole, bacteria lyses. Isn't that cool? It's pretty neat. Okay, so let's look at some of these chemical and physical defenses. If we're talking about the chemical and physical defenses, what are we at? 1, 2, or 3? 1, okay? So, let's start with up here, the sebaceous glands. What do the sebaceous glands release? Sebum, right? An oil, a waxy material. What is the pH of that waxy material? About 5. Yeah, about 5, pH 5. Okay? A lot of microbes don't like pH 5 except for the fungi, right? Okay? Now, tears and saliva, they have enzymes in there called lysozymes. Okay? And that helps to break down any invaders. Okay? We make mucus. Yay! Mucus is awesome. Where do we have mucus? Upper, okay. You are upper respiratory, okay. Yeah, here. And you also have a thin layer over the lungs, in the lungs. That thin layer is replaced every 20 minutes. Did you know that? It's fast when it's replaced. So that layer in your lungs is every 20 minutes. What happens when you're dehydrated? It takes longer and it's thicker and it doesn't work as well. That's why it's so important to be hydrated, because you're replacing that layer of mucus, that really fine layer of mucus. And what gets caught in mucus? Any debris, any bacteria, any viruses. And if it's in your upper respiratory tract, okay, what happens to that mucus? Okay, you either swallow it or spit it out. What? Hack it out. Yep, that's right, hacking. Okay, so yeah, you get it out with all of those things trapped in the mucus. And you'll say, but wait a minute, I shouldn't be swallowing it. Aha, what's the pH of this? 2. Anything that's in that mucus should be readily dissolved. The only exception is patients that are on protein pump inhibitors, right? Because then their pH won't be quite down to 2, okay? All right, we've got earwax, we've got sweat, we've got stomach acid, intestinal enzymes, and mucus, and we've got cilia. What are cilia again? Little hairs, and they move like windshield wipers really fast. Now, which population is going to have reduced ciliary action? Smokers, for sure. And it's not just the cilia here, it's the cilia everywhere. So we know they're inhaling something that's getting transported. So, yeah, okay, cool. So, skin, strong, thick barrier. Nasal hairs, okay, and cilia, all right? I hate to see everybody, you know, I've seen them in the, you know, like, oh, I'm going to get rid of all my nose hairs. No, don't do it. It's a protection. Trim them, but don't get, you know, they stick wax up here and then they pull them out. Yeah. I don't know. It just bothers me. And it's like, oh, it hurts. I'm like, yeah. Okay. Mucus, coating of cells that it stops attachment. Okay. You have the flushing effect, which is sweating, saliva, tears, mucus, urine, defecating, vomiting. That's a flushing effect. What are you doing? You're trying to flush those microbes out. Okay? Then we have blinking. Y'all blink. Yeah, that helps to get rid of microbes. Okay? You have coughing, sneezing, and the ciliated escalator. Okay, that moves microbes out. Did you learn about the ciliated escalator in anatomy? Okay, should have learned about that. That's important, not only for respiratory, but if you're female, the escalator is particularly strong, but that cilia motion is so important. It not only here, but digestive and reproductive and stuff. Okay, blood clots, seal off portal of entry, okay? So that's cool. And then damaged cells hopefully undergo a rapid repair. So these are all your physical barriers. Let's look for your chemical defenses. Okay, these are lysozymes that is in tears, sweat, and mucus, and it can digest peptidoglycans. Okay, saliva contains antimicrobials. That's why mom used it all the time, right? Oh, you got a cut? Let's lick it. Ugh. All right, sweat, pH, and high electrolytes. So when you sweat, you also put out salts, and that is also antimicrobial. Skin is coated, okay, in sebum, which we already talked about the pH of that. Okay. Skin has an acidic pH with all the microbiota. The gastric acid is secreted in the stomach. Digestive juices, enzyme and bile that you have in the GI. Okay, you also have, well, some of you have a vagina that is acidic, and the genitourinary, it has its own microbiota. And then, of course, microbiota prevents opportunistic infections, so it prevents overgrowth of some and prevents the growth of invaders. Okay, so that's a lot of chemical defenses. Let's look at what pictures this looks like. Okay, this is in rat mucosa, and you can see the lumen is here, and here's the mucosa. Okay, so you've got a thick layer of bacteria that between the surface of the lumen and the surface what is it? I can't even look this close. Okay. Where are we? Okay, yeah, the lumen and the mucosal surface. So look at that interface. Wow, that's jumping around crazy. Okay, and then if you look at the higher magnification, you can see what happens. So you take this, wow, look at that layer, okay? So where is most of your immune system, really? You know? It's in your gut, with gut association lymphatic tissue. It's called GALT, okay? Did you talk about this in 50B? Okay, all right. If you haven't talked about it in 50B, you might want to pick up your anatomy book and take a look at GALT. I mean, we'll talk about it a little bit, okay? We have ciliated mucous membranes that flush everything out. And you can see here the cilia, and they work like windshield wipers, and that's where they're located, right in here, okay? To help get things out, and you can either swallow it or cough it out or something. Okay, so we good so far there? Okay, so what we're gonna do is take out a paper, and I want you to write from memory without looking at your notes at all, okay? Without looking at your notes, see if you can do the first and second lines, things that we just talked about, the first and second line of the text. Try it all by yourself at first. We're going to try it. What can you remember on your own? Okay, take a look at what your partner has got and see what you missed.
And the microbiota also helps account for the pathogens and they like and sometimes they also help like lower
Okay, what did you come up for the first line? Skin. That should have been at the top of your list. What else? What else? What? Sweat. What does sweat have? Lysozyme. Who else has lysozyme? Tears and what else? Saliva. Okay, what else you got for first line? What is it? Coughing. Okay, what do we call that? Good, the flushing effect, yeah. Okay, what belongs with coughing? Sneezing, blinking, vomiting, diarrhea, anything, flushing everything out, right? Right? Okay, there we go. That's your interpretive dance. Okay, what else? Blood clotting. Wait a minute, how many of these are over here in the second line? There we go. Okay, there we go. All right, take a look at your notes. Take a look at your notes. Fix your chart. Which is first line, which is second line? Are you sure? Stomach acid. Yeah. Okay. What? Phagocytosis. And what cells do phagocytosis? Yeah, that's second though. Yeah. All right. What cells are we talking about? Natural killer cells, eosinophils, monocytes. Okay. Good. All right. Cool. Okay. We haven't really talked too much about the second line so far, have we? Oh. Okay. So, anything else out here that you didn't get that you may have had on there? What? Cilia. Oh yeah. Cilia. Okay. Cilia. What else?
Fever.
Fever. Is fever first line or second line?
second line.
Yeah, there we go. Okay. You like fever though. Okay. All right, cool. Okay, anything else? What? The normal microbiome? Yep, okay. So I'll just put micro B here, okay. Good. All right. So we essentially did a one-page test on everything that we've just done, which is cool. Okay, so let's move on to, okay. All right, what is a healthy immune system responsible for? It's responsible for surveillance, for recognition and destruction. Okay, and I want you to pay attention to these little cells that are over here. Part of surveillance and the key to surveillance is being able to recognize self versus non-self. Was this discussed in 50B? I hope so. Yeah, a little bit, okay? So you have these things called major histocompatibility complexes, okay? And what they do is they help, they're on the surface of the cells, and they help the immune system recognize what is self versus not. And they can tell from the major histocompatibility complexes. So for example, we've got markers on here. This is self. Okay, we're not going to have a reaction. All right? But if it's non-self, and you've got different markers, you don't have the MHCs, then you can target this white blood cell will target it for destruction. Yeah?
Is that what happens with type 1 diabetes?
Yes, I was just going to talk about that. What happens when the body stops being able to recognize self versus non-self? It attacks you, so that's your autoimmune diseases, type 1 diabetes. What's another autoimmune disease that uses self versus non-self? Lupus, yeah. Okay, there's some GI things that are type 1 major histocompatibility complex, where the self versus non-self. Now, here's the scary thing. When does this usually happen? Anytime. Okay? I want you to remember this because the book doesn't make a big deal about it, but this is really critical. Your immune system can change on a dime anytime. Okay? You can develop allergies at any age. You can develop an autoimmune disease at any age. And you'll say, why? Boy, if you could figure that out, that'd be cool. That'd be really cool. That's like the monster, that's a Nobel Prize if you can figure out some of this stuff, really? Okay? So, yeah, anytime. So always be aware. And, there was a journal article out today about how the measles virus, we talked about how it eliminates a lot of your memory cells and things like that. They're now calling it immune amnesia. Okay, where your immune system can't even remember what was that. My prediction, just as being, thinking about this, if it can do that for immune system, could it do it for self versus non-self? Oh yeah. This is a little protein you have to wipe out. No big deal. So yeah, think about that. Okay, so what are your immune parts? There's blood, lymph, RES, ECF. What is ECF? Extracellular fluid. Okay. And GALT, which we already talked about. Okay. So this is your components of your immune system that is constantly working to protect you. Okay. Let's talk about phagocytosis a bit. Okay. So we already talked about non-self versus self, and that's done through MHCs, major histocompatibility complexes. Okay. We also have things that are called PRRs, which are pathogen recognition receptors in host cells. And they use what are called TLRs. I know, this is a whole new vocabulary. Toll-like receptors to recognize the pathogen. How do they do this? They do this with surveillance. I have tried to find a good talk that would talk about these toll-like receptors, And everything starts out in 5 minutes, it's great, and then it goes way above what we're doing. So I'm still trying to find something that will help you visualize what's going on, okay? Then the other thing, so if I'll keep you posted. The other thing that pathogens have are PAMs, which are pathogen-associated molecular proteins, and this is used in recognition. So you can't see this going on, but you've got a whole host of different kinds of receptors and proteins that are actively surveilling and part of your surveillance system. So phagocytosis can happen. So these things can be chewed up. So we have phagocytosis, we have cytokines are released. What are cytokines? This is the first time you're seeing cytokines. What are cytokines? They are chemicals that are released by immune cells. There's about 25 of them, cytokines. And what do? Well, some of them promote inflammation, some of them help to calm down inflammation, okay? And they all have different names, and they usually start with IL, okay? So there's 2, IL-2 alpha, there's 13, 15, 5, 10, they all have a number, okay? Those are interleukins. Those are the cytokines. Now, if you want to see what happens when cytokines get out of control, look at people that got COVID. One of the things the COVID virus did is not only stimulate the cytokines, but then confuse them. So when you look at people that died from COVID, it wasn't the virus itself, it was from something called a cytokine storm. And what would happen? Well, cytokines come in and they help to clear out, debris, they signal things so debris, damaged tissue can clear out, but they're supposed to be shut off. In COVID, they got confused. And so they damaged healthy tissue way more than should have happened. But that's one of the things this virus did, okay? So it wasn't just, it's a COVID virus. It was the immune response to the COVID virus via the cytokines. So when we first started seeing COVID patients, we're like, okay, well, we're going to treat them like they have a virus. Oh, and then when they got too sick, they're like, well, we want to give steroids because we can see the immune system ramping up, but we knew that would hurt their fighting the virus. You see the problem? That was a big problem. And we're still 20 days, 18 days out from good antibodies. So yeah, it was a problem, okay? But yeah, when all of this is going on and it's recognizing these things, you can have these, all of these proteins and chemicals working to stimulate, and then later on in the response, suppress the immune system. Okay, got 2 minutes. Okay, this talks about self versus non-self. MHC markers are found on every cell surface except for what? Red blood cells, right, except for red blood cells, okay? MHC1, there are different MHCs. There's MHC1 that's critical for self and non-self. There's MHC2, MHC3, okay? But this is how it works. If the markers are there, the white blood cell says, oh, this is me, I'm not going to do it. And if the markers are not there, the MHC1 is not there, then you get PRRs that are made by the white blood cell, and you get pathogen markers. These are the PAMs, the pathogen markers, okay? And you get destruction. But those MHC1s are critical. Now, before we stop for today, I want to talk about the overview of blood products, okay? How many of you have had 50B? Okay, if you've had 50B, this is something that I hope was covered. Is this covered? Okay, you got to do a review on this because you need to know, where do these cells come from, all right? Where do these cells come from in a healthy immune system? Okay, and that's what 50B is, and then we'll screw it up in micro, right? Okay, so make sure you know which are myeloid derived and which are lymphoid derived, and where do they mature? All of that good stuff. Good review for Wednesday, okay? Is that good? Awesome, okay.